I'm Joe Grabowski from the University of Pittsburgh and representing the Pittsburgh Conference for this 2014 Pittsburgh Conference Achievement Award Symposium in honor of Ben Garcia. The Society for Analytical Chemists of Pittsburgh, so-called SACP, is the sponsor of the Pittsburgh Conference Achievement Award. The award, presented annually at Pittcon, recognizes individuals for outstanding achievements in analytical chemistry or applied spectroscopy, work accomplished within 10 years of the receipt of the PhD. Ben Garcia earned his bachelor's degree in 2000 from the University of California at Davis, where he worked with Carlito Labrilla, and I guess spent one summer with Jack Beecham. Was it at NSFRU? So connection there. Then from Davis, from California, Davis decided to come out to the east and went to earn his PhD at the University of Virginia in 2005, working with Don Hunt. 2006 to 2008, Ben was an NIH postdoctoral fellow at the Institute for Genomic Biology at the University of Illinois, where he worked with Neil Kellerer and Craig Mizzen. In 2008, he started his academic career at Princeton University in molecular biology with an associate appointment in chemistry. In 2009, he added an appointment as a core faculty for the quantitative and computational biology program at Princeton, and in 2010 became an associate member of the Cancer Institute of New Jersey. In 2012, he accepted a position as the Presidential Associate Professor, Department of Biochemistry and Biophysics at the University of Pennsylvania Perelman School of Medicine, joint with appointments as a faculty member in the Epigenetics program, where he also serves as a Director of Quantitative Proteomics. Ben has secondary appointments in the Department of Chemistry and the Department of Bioengineering. As you might guess from a rising star, Ben has received a number of awards, just a few. In 2012, he was an Alfred Sloan Research Fellow. In 2011, he was awarded a Presidential Early Career Award for Scientists and Engineers, the so-called PCASE Award. In 2010, he received an NIH Director's New Innovator Award, the Agilent Technologies Thought Leader Award, and the National Science Foundation Career Award. In 2009, just at the start of his career, he received the American Society Mass Spectrometry Research Award. As you'll hear in a few minutes, Ben is focusing much of his research efforts on developing proteomic techniques to better understand chromatin structure, and for that work is one of the reasons that he was selected for this award. I'm going to turn the podium over now to Heather Joswa, the chair of the Society for Analytical Chemists of Pittsburgh.
Thanks, Joe. Welcome, everyone. On behalf of the Society for Analytical Chemists of Pittsburgh, I would like to congratulate Benjamin Garcia for this year's Pittcon Achievement Award. If you can come up and receive your award. I'll say what I'm telling him. His plaque stayed in Pittsburgh, so this is a plaque for tomorrow. It's going to look exactly like this, but with his name on it. Congratulations. Yeah.
So we're a little bit ahead of schedule, but we're going to go ahead and take advantage of that, giving Ben perhaps a few extra moments. So Ben is going to talk about mass spec approaches to quantitatively address the dynamics of post-translational modifications to histone proteins. Ben, thanks.
Well, first, just want to say a few thank yous to everybody involved before I kind of get into the science. So I really do want to thank speakers who took time out of their busy schedules to be a part of this. Everybody that is a part of this session has, in one way or the other, kind of indirectly or directly influenced my career in different ways. I've been a big fan in following the research of the Broad Belt and Wysocki groups for a long time, since I was an undergrad, actually, in Carlito Labria's lab. Scott Gerber is somebody who I kind of really respect for taking a really quantitative analytical chemist approach to solving some really, really difficult problems in biology. And I remember reading his aqua paper, I guess it's over 10 years ago, something like that, Scott, 11 years ago. And just thinking, wow, things just really got quantitative all of a sudden. Jack Beecham has been a great mentor, friend, everything over the years. And he's still somebody that I occasionally bother for things and that collaborate with a little bit. And, he gave me an opportunity to come into his lab in the summer and really, see what science at a big university was like, and was the person that actually told me that, well, you want to study proteins, and this was before proteomics became a word. He's like, you probably should head east to Charlottesville. And that's where I joined Don Hunt's lab. The people at the bottom here are what I would consider my research mentors throughout the years and different phases of my career. Carlito Labria really, I wouldn't be here if it wasn't for him. He kind of took a chance on me and really took me in and gave me a lot of opportunities that I think a lot of undergrads don't get, and a lot of responsibilities to be a functioning member of the group is equal to anybody in the lab. Don Hunt is just an unbelievable scientist, and I learned so much from being in his lab. It was just an unbelievable time with a lot of innovation and discoveries, and he's just an incredible individual that has been really supportive of me over the years. My postdoc with Neil Kelleher was an amazing time, probably the most fun time I've had, in my academic career. It was, a pleasure to be in his group. You guys know Neil, so imagine having him on a daily basis. It was just a really, really fun time. He's just an incredible scientist and such an enthusiastic guy and motivator. And one last slide before I get into the research again. I've just over the years since I started my independent group at Princeton University just had an amazing, despite what some of these pictures may depict, I've had just an incredibly intelligent group of highly dedicated workers who have just at many times just amazed me with how great they have been. Many of these people are off, establishing their own labs, such as Nick Young and others. And it's just been a pleasure to be associated with all of these people. And I definitely would not be here if it wasn't for them as well. So thank you very much again to the Society for Analytical Chemists, as well as Pittcon. It's really an honor to be included with all the past award winners of this achievement award. Okay, I guess I should get into some science. So my lab has, for a long time, and even this goes back quite a bit to even my graduate school days. I've been very interested in analyzing histone post-translational modifications, and there's a lot of different modifications that occur on these histones. What you're seeing here is just kind of the more abundant ones. I think this timer is off, so yeah. All right, just tell me when. And what you're seeing here is just the core histones, and you have 2 copies of each that complex together with about 146 base pair of DNA to make a nucleosome, the basic repeating unit of chromatin. And the most abundant modifications on the histones are probably methylation, acetylation, phosphorylation, although there's been so many new modifications that have been discovered with mass spectrometry and other approaches. These modifications are not static, and they have an immense amount of biological function, mostly regulating transcriptional activation or silencing, but also involved in various other processes such as DNA damage and even apoptosis and other things. And it's because you have these modification patterns on these proteins that can control gene expression outside of DNA sequence. These modifications make up kind of these epigenetic mechanisms. And, in attempts to try to understand this, there's been a lot of theories and hypotheses put forward, but one of these is the histone code hypothesis that states that these modifications might act as some kind of language or code on top of the genetic code. And when you think of a language or code, there has to be some way to interpret this language or code And the way to do that is that there's specialized proteins that have distinct domains that bind specific modification sites. And this is the first step of recognizing this code, and this elicits further downstream events. So right now it's a really incredible time to be in this field because there's a lot of discoveries to be made. There's people looking at combinatorial modification patterns and what does that mean, as well as just a lot of biological function that needs to be discovered and attributed to these modifications. My group has been working hard on developing a lot of mass spectrometry-based approaches to tackle various issues with regards to these histone modifications. And I won't talk about most of these, but we've been working on very quantitative approaches to look at multiple cellular states and determine which modification patterns are changing from state A to state B. We've been working as well on looking at intact proteins or very large stretches of the histones to try to understand this kind of combinatorial histone modification pattern, as well as linking genomic type of experiments to our proteomics data so that we can kind of read the chromatin landscape and see what histone modification patterns are found on distinct regions. But what I'm going to talk about today is an area that I believe is obviously important for histone modification dynamics, but also in general is probably the next kind of aspect of looking at protein modifications in proteomics in general. And that is trying to get at the kinetics or flux of modifications. How fast are they put on or off? And we've been working with regards to understanding histone modifications like methylation and acetylation, how fast they're getting generated and removed from various different processes. But also now we're extending it to other classes of modified proteins as well. And this all kind of had come from the question that we often got from various collaborators. If I did an analysis, state A and state B, and I found that lysine dimethylation was increasing from state A to state B, well, the first question that I would get from a collaborator is, well, does that mean that the monomethyl pools are increasing to increase the dimethyl pool, or are we seeing a demethylation event where we're taking a trimethyl and removing a methyl to increase the dimethylation event? And this was a kind of very difficult question to answer with kind of the conventional proteomic approaches. We can kind of infer things and, sort of get at it, but we really didn't have hard evidence. We really couldn't get at how the flux of these modification patterns were changing from one way to the other. So we kind of thought about it, and there's a lot of hints in the literature about how to do this as well. But for this, we kind of turned to dynamic SILAC, as some people call it, or pulse SILAC, pulse chase SILAC. So for those who may not know what SILAC is, it's the stable isotope of, labeling of amino acids in cell culture. And the normal way that most researchers use SILAC is for comparative quantitative proteomics, where you can take state A, you have your cells. It's a metabolic labeling approach. You can take one set of cells, label them in the normal media, so it has normal amino acids, normal stable isotope, C12 and 14 amino acids, and you have a second set where you'll have some kind of condition, treatment, or perturbation, and these cells are grown in heavy media, which has heavy C13 and N15 amino acids. After several doublings, extract out the proteins, mix them together, and when you run it in the mass spectrometer, you can do relative quantification because you know the signal that's coming from the light and the heavy from each sample. An offshoot of this is that people have been using SILAC in more pulse mode to look at protein turnover. So you could take your set of cells, grow them in the normal media, transfer them to the heavy media so they start getting labeled, but you don't wait until they're fully labeled. You start taking time points along the way. Or you could do a traditional pulse chase type of experiment. And what you should be seeing here is degradation of the old protein and synthesis of the new. And there's been some really nice large-scale analyses of all kinds of different proteins, even from in vivo mice and things like this. However, if we're looking at histone modifications, specifically methylation, we really wanted to monitor how fast the modification was changing or being turned over, and not really the protein turnover. So we thought about it for a while, and we've come up with different strategies to be able to label the newly modified histones and proteins in cells. So if you take a look at the biosynthetic pathway for methylation, all the proteins that get methylated, or actually large majority of methylation on biomolecules, is coming through the pathway where methionine is converted to S-adenosylmethionine, which is the cofactor that is being used by many methyltransferases, practically all methyltransferases. And we felt if we can use a heavy labeled methionine, we should be labeling all the proteins that have a methionine. That's fine. And that's good because it's kind of like a background for us to see how fast the protein turns over. But at the same time, all the proteins that are getting methylated, we should be able to label those as well. So grow the cells and label methionine. And what we should be seeing is decrease of the old methyl, synthesis of the new methyl. And that's actually what you see when you start looking at these experiments. Here is data from lysine mono, di, and trimethylation. And this is over several days. Methylation is really, really stable, old, and then the new. And you see monomethylation decreasing, dimethylation, or monomethylation, the new, coming up, and then dimethylation decreasing, trimethylation decreasing, and then you see the newly synthesized species coming up. For the di and the trimethyl, though, you see some intermediate species that have an old label and a new label, and then, you know, combinations of 2 old labels and 1 old label with new label. And this kind of confused us for a little bit, but what we found, if we take this example, is that this species right here was a dimethylation that's decreasing, already existing. Here's a dimethylation increasing, so this is a species of histone that was previously unmodified that then got dimethylated. And this species right here is something that had 1 monomethyl and then got processively methylated during the experiment. So this is a species that existed as a monomethyl and went to dimethyl. And over here we see all the different species for the trimethyl, the 0 to 3, 1 to 3, and 2 to 3 conversions. And, you know, you might not think, well, you know, is that really important? I mean, they're all trimethyl, all dimethyl. But it's actually really an important feature of this type of work because, you know, before we can tell if dimethylation was increasing with kind of this unlabeled silac or unlabeled approaches and not the silac approach. But with a little bit of mathematical modeling and being able to monitor all these different states, if we actually see dimethylation increasing and it's this species, we can actually determine the rate kinetics and find out if it's a trimethylation event that's getting demethylated or where exactly in these pathways the methylations are increasing. So it gives us a lot of specificity. And there's various enzymes that have really specific substrate requirements. So if we know if it's a 1 to 2 conversion or you know, a 0 to 2 conversion, there's actually different enzymes that do that. So by monitoring all these different states, we can actually not only determine which pathways were activated to get to those final epigenetic states, but we can also determine the enzymes responsible. And we've been able to use this approach to monitor the half-lives of large majority of histone methylation patterns, just some shown here. As you can see, the methylation patterns are actually quite slow in most cases, not all cases. In general, silencing marks such as the K9 di and trimethyl are very slow. So you see the protein half-life right here, and then di and trimethyl are either right at the half-life or slower, monomethyl much quicker. However, if you look at a mark like K36, mono and dimethyl, they're both pretty fast. So it depends on where the modification is. If it's involved in gene activation or gene silencing, that seems to really regulate the turnover, and that probably has something to do with the genomic regions and the accessibility of enzymes to turn over these modifications. So we've been using this type of labeling approach in various different types of experiments to try to understand different aspects of histone biology. And one of the questions that has kind of eluded chromatin biologists for a long time is, how are these modification patterns inherited? When you talk about epigenetics, it means that these modification patterns are inherited from one cell generation to the next. These gene expression patterns and the histone modification patterns associated with them are, established in the same genomic regions from, the parent cells to the daughter cells. But, there hasn't been a very quantitative look about how these modification patterns are established. And the whole kind of thought in the field was that, histones come into S phase unmodified. They come in naked, they're not modified. And it's thought that before you get to mitosis, or shortly after S phase, but definitely before you get to mitosis, the modification patterns on these histones have to be firmly established because the cell is going to split at that time. So we decided that maybe we can take a look at the modification patterns of different histones, using cell synchronization and this metabolic labeling approach to specifically label the methylated proteins to try to get at this question of when are these, and we had a specific interest in this modification site, K27, when are these methylation patterns established? And like I said, our kind of initial hypothesis, because of all the thoughts in the field, was that these modification patterns have to be restored. The new histone that comes in has to look like the old histone shortly after assays before mitosis. So this is kind of the model, and we want to kind of test, well, when the new nucleosomes come in, nothing there, when do the modification patterns, when can you get to the point where you can't really distinguish the old and the new in terms of their modification patterns? So we turn to this, again, this stable isotope labeling of amino acids in cell culture approach with the label methionine, which I talked about before, using cell synchronization. So the cells are going to be synchronized in G1S. And a little bit difficult to look at this data when you're first staring at this, but what you're seeing here, here's the general protein turnover. So this is like, and here's the color code for the different phases of the cell cycle. So we've synchronized them here. The new histones are coming in right here, and they're getting slowly, some of them degraded, and many of them modified right here. So this is K27 monomethyl, and then K27 dimethyl. And I'll kind of run you through a little bit, but 1,0 is an already established monomethyl, so the 0 means old, and 1,1 is a monomethyl with the 1 being new. So what you're seeing is that over time, the old methyl, is getting turned over as you would kind of expect through the cell cycle. And what we're keeping an eye on here is this area right here, this G2M phase, because this is mitosis. So this is where we believe the modification pattern should be fully established before the cell split. And for K27 monomethyl, it's definitely the case. The methylation patterns increase quite a bit, and it's higher than the protein turnover rate. So we're pretty happy that that is happening. However, when you get to here, to the K27, you see that there's definitely kind of a lag between the proteins being synthesized and turned over, as well as the modification patterns. So the old is kind of decreasing. And here in this it's kind of hard to see here, but here's the kind of, I guess orange is the protein synthesis rate. And what you're seeing here is the new methyl, so the the 2,2 is the new dimethylation event. And as you can see here, this methylation levels don't reach the protein synthesis levels until way past mitosis. And that's kind of, a little bit intriguing and head scratching. But what we can determine is that monomethyl is mostly, coming up due to turnover. Dimethylation is looking like it's coming from, two different types of synthesis pathways. But bottom line is that the modification patterns don't seem to reach the levels of the newly synthesized proteins until way after mitosis. And it's even more pronounced for the trimethyl. So here's the protein synthesis rate. And we have a lot of different species, but if we even add them up all together, so we had those going from 1 to 3, 0 to 3, 2 to 3 methyls. If we add them up all together, we still don't come close at all to reaching the newly synthesis rate. And we don't reach the synthesis rate until way after mitosis. So here's just different biological replicates for this K27 trimethyl. And we did a different synchronization to be able to get these far time points and get past mitosis, because with the synchronization we did before, once it gets past mitosis, the cells start becoming unsynchronized. It's harder to control. But if we do a different cell cycle block, we can get really far points. And what we find for the trimethyl is that it really doesn't come close to the protein synthesis rate until way after mitosis, probably to the next G1 phase. So this is quite kind of unexpected, but what this tells us is that in S phase, histones are coming in unmodified, they get monomethylated, and this seems to be the really important event. Once you establish a monomethyl there, there will be, the propagation to get it to the trimethyl, but this won't occur until the next G1 phase. So it does not have to happen before the cells split. So in a sense, you could think that these epigenetic histone modifications might not be epigenetic after all. And there's been some really nice papers by other groups showing that the modification patterns themselves are probably not epigenetic, but the enzymes are. So the enzymes come in and are established in S phase, and they stay on that chromatin, and somehow they're being targeted to the specific genomic sequences. And they are probably the epigenetic drivers because the modifications show up, hours later. So this is kind of just one nice application of what we can do. I'll talk a little bit about a few other applications to different post-translational modifications, some histone, some not. We're going to see some really nice research from Scott Gerber later, who's going to talk about phosphoproteomics and its involvement in various cancers. I don't think I need to overstate this. Phosphorylation is an important post-translational modification. I think this is probably even higher now. Probably close to 50% of all proteins or something like that are known to become phosphorylated during, some time or another during their lifetime. And this phosphorylation, obviously regulates many different areas of biology, too many to name here. What is known in various cases is that the sites of modification and the duration and the magnitude of signaling influence the biological output. However, that's, this duration and magnitude are kind of difficult to capture. So we've been thinking about how to do this. How do we capture newly phosphorylated events and see how long these phosphorylation events their turnover, or how long it takes for these phosphorylation events to completely finish their function. For this, we also use a different labeling approach, which we kind of learned called SILAP, but it's probably not going to catch on, I think. And here we take cells and permeate them and introduce O18 ATP into the cells. Or we can also take nuclei if we specifically want to look at chromatin. That's kind of some of the data I'll show you. So if we take these cells or nuclei that we've gotten ATP in there, kinase is going to come in, put this new ATP onto a protein, and we'll be able to monitor all the proteins that get phosphorylated because what we should be seeing is there's always going to be a background level of old phosphorylation, but any new phosphorylation event is going to be a 6-Dalton mass shift. So this should increase the confidence in the phosphopeptide identification to have this kind of extra species that we're able to monitor by mass spec. Obviously, we're able to distinguish old and new phosphorylation, so you can imagine using this in various conditions to see how fast proteins get phosphorylated. And what we find is that oftentimes with at least our phosphoproteomic data, unless we see like a really huge change and we do incredible amount of replicates, it's hard to determine something that changes by 10% or less. I mean, it's just really, really difficult to get that precision. So if you have time point 1, time point 2, and you want to see if it changes, but it's only changing like 10% or less, it's really hard to capture that. However, with this approach, what we feel it should be a little bit easier to capture those events because time 1, we'll have all the background level phosphorylation. Time 2, even if it's small, we should be able to monitor these new species. And I do have to say, we just got this accepted into MCP, so it's in press. I think I told you all that before. So here's just the workflow for how we started this. We either take intact cells, permeate them, and add the ATP, or in this case, we're isolating nuclei, and this has been done for decades by chromatin biologists in nucleo type of experiments. We add in the ATP, take time points, go through the titanium dioxide enrichments and mass spectrometry experiments to identify the phosphorylation events. And we monitor by metabolomics the amount of ATP steady state levels that we see. There is some interesting chemistry that's going on there with like back exchange and things like that. But for the most part, we do see the labeled species the most. And here's kind of what some of the data looks like. So some time point later, we see the species. Obviously, it's the change. And then we look at the MSMS, as you can see here, if you're monitoring, some of these particular ions, such as this Y4, it's in the light, 536, and the heavy, 542. We've created a whole pipeline to be able to look at this data. Most commercial software just kind of can't take it. So we've kind of worked on that in collaboration with Jonathan Goya at Princeton. And here's just some of the kind of raw data and curves that we're getting. I'm going to speed up a little bit, but because we're able to get these time points over time, and we're seeing these new methylation events, we can actually get at the rate constants of phosphorylation on a kind of a global scale. And here's just an example of, you have many different rates. You have some that are a little bit slower, some that are much faster, and you can even see this visually in the mass spectra for this transcription factor versus this protein if you're looking at the newly phosphorylated species. Here's 2 sites on the same protein, that same transcription factor. As you can see, really rapid site here being phosphorylated. However, this site is very slow, low level, the kinetics are much lagging behind this. And this could be, it could be an artifact of the way we're doing the experiment. Maybe this phosphorylation site doesn't happen for whatever reason readily in the nucleus, or maybe the kinase responsible here is a little bit more active in the nucleus. But at any rate, we can pick up these differences by mass spectrometry, and we can even pick up intermediate species like we did before with the methylation. So you have 2 phosphorylation events on this protein. I have no idea what it is. But you can see that you have the old methylation, you can have processive or old phosphorylation, processive phosphorylation, or newly completely phosphorylated events, 2 phosphorylated events. And, obviously we're doing phosphoproteomics, so we can do this in a large scale. We're not doing it one at a time like most biologists have done over the years, but we're able to get these rates over time, so we have these clusters of slow, mediate, and fast turnover. And when you kind of look at this and the distribution of the biological function behind some of these, a little surprising, but those involved in metabolism are the fastest to be phosphorylated. So it's kind of interesting. And we can see by looking at the kinases, which kinases are more active, in this process. As you can see here, these blue and green clusters are the fast ones. So all these proteins right here are really fast and rapidly induce phosphorylation. We can use this in comparative mode to compare the phosphorylation rates of various different processes. So you can see here, if we compared cell cycle phosphorylation, G phase to M phase, sometimes we see things that are faster in G phase, some things are faster in M phase. So these kind of experiments can be coupled with many different types of perturbations, kinase inhibitors, all kinds of different things to look at the rates of phosphorylation. We also look at, I guess in the last few minutes, I just want to talk about a few more things that we're starting to get a handle on, other types of modifications. So what we're also interested in is protein acetylation, and we know that we can use labeled glucose. Glucose will get converted to acetyl-CoA, and that will contribute to both fatty acid synthesis as well as protein acetylation. And it doesn't show up really well here, but by looking at the different pathways that can contribute to acetyl-CoA, we see that glucose by far is contributing here. It's actually, you can see it a lot better right here. So if we put in labeled, universally labeled C13 glucose, what we should be seeing is 2 carbons being added by this acetyl group onto a lysine molecule. So this is a doubly charged peptide. We should be seeing this species right here increase, and that's exactly what you see over time. And acetylation is really fast. Most half-lives are probably around, 30 minutes or so, something like that. So much, much faster than methylation, which was very stable in hours. We're also starting to look at proteins that have sugars put on them, O-glucanac modifications. So it's obviously an important modification that affects various different types of biological processes and disease states such as cancer. And we're starting to look at the dynamics of O-glucanac, specifically in chromatin biology. It's an interesting modification that has many implications for gene transcription. It's been found on histones, and we see it on histones as well in various processes. But we can do the same thing, label cells with labeled glucose and kind of follow during glycolysis in the TCA pathway the distribution of glucose, and it finally converts to acetyl-CoA, but which we used for the acetylation dynamics. But also, obviously, then becomes the glucanac donor, and we were able to see the glucanac turnover, old and new as well, and it's incredibly fast, and we're trying to get better at capturing it and measuring it. Last thing I'll touch on, and then I should be done, is that we're also very interested in which other proteins get methylated in the cell besides histones. There's about 50 enzymes that are part of this human lysine methyltransferase family. They're really kind of an interesting class because only about half of them are known to work on histones. The other half, people don't know what they work on. And even many of the histone ones are known to have non-histone substrates. So the question is, how many non-histone substrates are there in the cell? And there is a lot of interesting biology behind them as well, because many of these have been linked to various cancers. The most famous one is MLL, this mixed leukemia lineage protein, which has a lot of well-defined genetics for how this protein's activity is altered, either overexpression downregulation of its activity. But several other of these methyltransferases are also linked to many different cancers, and they're the really hot target right now in the pharmaceutical industry for the next drug space, this epigenetic druggable targets. Cancers are mostly genetic in nature, but because of the sequencing data that's coming out, many of these genetic abnormalities, whatever they are, chromosome duplications or amplifications, these different types of things, many of them are affecting epigenetic enzymes and associated proteins, many of these methyltransferases. So we're developing methods to look at non-histone protein lysine methylation dynamics. We have developed several panels of highly specific antibodies that took about 3 or 4 years to really get high quality antibodies where they're very specific and general, so that the antibodies will only recognize monomethyl, dimethyl, or trimethyl. And we've coupled these with mass spectrometry to produce the largest catalog of non-histone methylated proteins to date, which we published last year, over 500 non-histone methylated proteins. And now with second generation antibodies and different better mass spectrometry, we're up to about 1,400 now non-histone methylated proteins. And we're starting to define the methylomes of very specific enzymes. So we can go in and use inhibitors. This UNC molecule is an inhibitor to the G9A enzyme. So minus inhibitor, plus inhibitor. We can do quantitative proteomics when we do the methylation labeling in combination with other labeling, and see that this protein was identified as Wiz. It's methylated at this lysine residue, and it's decreasing upon an inhibitor treatment. And we've also are coupling these with knockdowns of various enzymes as well. So we expect to define the non-histone methylome of all of the major methyltransferases, especially those involved in cancers. So in summary, once again, thank you for this incredible honor. It's really, just an incredible feeling, and, just a really a great kind of achievement, not just for myself, but for my research group, who's worked so hard to really establish themselves and, try to contribute to various aspects of biological research. And just like I kind of touched on today, we're really interested in how fast these modifications are getting distributed onto various proteins, whether it's histone or non-histone, and we're starting to attack various different types of modifications, methylations, acetylations, phosphorylations, and as you can see, other things as well, and we have other things in the pipeline already as well to go after different classes of modified proteins. But I think this is kind of the missing key point of proteomics work, is the kind of temporal aspects, looking at these type of kinetics and asking what that means. So again, just want to thank all of, this is actually the current group, but obviously the past and current group have all contributed incredibly, and built upon each other's successes and continue to amaze me. Great cast of postdocs and graduate students and staff and collaborators, and able to get funding from a lot of different sources. And I'm really thankful that I've been able to start my career at Princeton. They gave me a chance when I probably didn't deserve it. And thankful for Penn and all the support I get at the university. So once again, thanks. I'll try to answer any questions if there's time. If not, I'll probably stick around for a little bit and can talk to some people at the break. Thank you.